Hello, everybody. My name is Abdul Aziz Hatko. I am a computer science student, and this is my bachelor thesis. It will talk about the higher order adaptive dynamical system modeling of the role of epigenetics in asthma. So, as an overview, we will talk first about what asthma is and which type of asthma the paper focuses on, and then the models will be presented. After that, the simulations will also be presented, and then a conclusion and future work will be discussed. So, what is asthma? Well, asthma is a common and complex respiratory disease. It is characterized by the inflammation of the airways and other symptoms. And this paper focuses on the asthma type called type 2 high asthma, which is basically allergic asthma. So the other type is type 2 low asthma, which is essentially the non-allergic. And then for the connection to the epigenetics, it will look at the methylation of the IFN gamma gene, which is responsible for creating interferon gamma, which is important for the regulation of the asthma mechanism. And if this is not well regulated, then asthma symptoms will occur. So its methylation and its downregulated expression will lead to worse asthma symptoms. And there will be a comparison between the normal and the extreme case in the following simulations. Okay, so for the models, there will be two models. The first model will create a asthma pathophysiology. So it explains how asthma works from the moment that there's an inhalation of allergens and how this mechanism can fail because of methylation of the IFNG gene. And then the second case will look at the hypothetical therapy, which is a therapy based on the epigenetic level. And it will be possible to see how these work. So for the combination functions that are used, 3 combination functions are used. So the alogistic combination function is used for mostly all of the states in the network model, and these are the biological states basically. For the step mod, it's used for one state which is representative of breathing under normal circumstances. So this just is a repeated duration, and it just goes up and down. So it just like represents different type of stimulus. And then for step ones, 2 states will use this. And what step ones does is it has a start time where it will go up, and then at the end time it will go back down. And the extreme case and the epigenetic therapy case will both use step ones. Alright, so this is the network model. And as you can see here at the base level, this is the asthma pathophysiology. So what happens here is this is the basic normal breathing state, and it has a direct connection. These arrows are positive if they're solid, and if they're dotted, it means that they have a negative effect on the other state. So this has a positive effect on the epithelial cell response, so it will make it more responsive. And then this connects to the dendritic cells response, which will lead to the Th0 cell response, which is the start of the differentiation of the Th1, Th2 balance. And this Th1, Th2 balance is important because if there's no imbalance there, or rather if there's a skewedness towards the Th2 cell response, then that means that the asthma response will be happening. So as you can see here, there's this regulatory cycle between each other, and also you can see that the Th1 and the Th2 both have connections that weaken each other. So they both try to suppress each other, and this is why the IFN gamma gene can suppress the Th2 response. So to continue, So the Th2 response and with the help of ILC2 cells both create cytokines called type 2 cytokines, and these cytokines lead to symptoms which are split here into symptoms that attenuate, so it makes asthma less worse. So for example, think about the creation of mucus that makes the epithelial cell less sensitive, while there are other symptoms that amplify asthma, so they make it more sensitive, like broncho hyperresponsiveness. And as you can see here, this one is the extreme case. So think about extreme allergen exposure. And what happens here is there's a line that goes all the way to the epigenetic level, and it affects this W state. And these W states are states that help the model or helps the model network self-model itself. And these context states are just for the context of each level. This connection between the context extreme to all the way to the top layer of the epigenetics, this is basically the methylation of the IFN G gene, and this methylation will lead into a lowered expression of the gene here. So this is basically a downregulation, and as you can see, it cascades all the way downwards into the immune system level, and here the connection between Th1 cells and the Th0 cells is weakened, which implies that the Th1 cells will not be as active as they're supposed to be. Alright, so this is the next model with the therapy case. As you can see, it's not different from the first one, except for a therapeutic state at the top, and this therapeutic state helps alleviate the methylation, so it intervenes and tries to unmethylate the IFN G gene. Okay, so we will look at the simulation overview. So there are two simulations. One simulation will be about the asthma pathophysiology without the therapy, and the other one will look at the one with the therapy. So this is the first scenario. This scenario has no therapy case. So we can split this plot in three parts. You can see that this is the first part, and then the second part is here. This is where the extreme allergen exposure is turned on, and then it turns off, and this is the third part. So what happens after? So in the beginning, you can see these two just going up and down, but like not really being very high level responsive, and these two are basically the epithelial cell response and the dendritic cell response. And everything else which leads to the asthma symptoms is just basically at a very low level, because there's no real sensitization and extreme exposure. All the W states also begin at the top, and once the extreme case starts going up all the way to 1, you can see that all these W states fall down, and this is the down regulation. At the same time, you can see that the epithelial cell is sensitized, and everything else is also being sensitized, and this leads to the starting of the differentiation of the Th2 response. And you can see here that this one that goes up and then goes back down, this is the Th1 response. So you can see that it fails at its regulation and it goes back down, while the Th2 goes all the way up, and this leads to the symptoms of asthma to also go up. And after the extreme exposure goes away, you can see that the epithelial cell response and its dendritic response are still like high. So you can see that they're also now more sensitized, and the symptoms didn't really go away, and the W states didn't really go up again. So this implies that after the extreme allergen sensitization, these symptoms remained. So let's look at the case with the therapy. So for the therapy case, it's still the same with the first part and the second part and the third, but there are two other parts added right now. So this is the therapeutic case, and then after therapy, what happens? So the first three parts are not that different. They're actually not different, and the fourth part where the therapy goes up, you can see that all the W states go up, and you can notice that because of them going up, you can see that the Th1 response also starts going up, and this leads to the regulation going back to normal. And this regulation leads to the symptoms going down, and also the Th1 response to go down. And after the therapy goes away, you can see that the epithelial cell sensitization along with the dendritic cells is also back to normal, basically. And slowly but surely, the Th1 and the response of the Th0 also go down. And this is because there is a regulation happening back, and it's not like dysregulated anymore. Okay, so now we can conclude that the methylation of the IFNG gene hampers Th1, Th2 regulation, which will lead to worse asthma response and symptoms. We can also say that the symptoms remain after the extreme exposure goes away, and the sensitization still remains after it. And then finally, the hypothetical epigenetic based treatment can help bring back the balance of the immune system and the mechanism that is responsible for regulating the asthma pathophysiology, bringing it back to where it was supposed to be. And for future work, it is very important to verify the model and the simulation with real world metrics. So the validity and the verification is very important to continue working with this model. Furthermore, the model can be expanded. This model has been abstracted to be to be important for the context of the gene that was looked at, but because asthma is such a complex disease, it is very possible to expand on this model in many different ways. And finally, it is also possible to explore the other genes that relate to asthma and other gene loci that lead to asthma symptoms with this model. Thank you guys for listening, and this was my presentation.